Okay, so here I'm going to discuss diversity in your antibodies, your immunoglobulins, and how this comes about. And so one thing to consider about antibody diversity is it's occurring at the level of your genes and how remember your genes go to RNA, goes to proteins, and your proteins are antibodies. And so how those proteins are folded and how their variable regions are generated makes an antibody able to bind to a diverse range of pathogens that you can come in contact with for your lifespan. And so diversity really starts in the germline. So what genes for making antibodies did you get from your maternal and paternal? And these genes are going to be codominant. Now the germline is going to give you heavy and light chain genes. And remember, the shape of your antibody is your heavy chains are going to be sort of on the inside, and they're going to go all the way through the trunk, and your light chains are only on the outside, and they're shorter. So these are your light and your heavy. Now that is happening at the level of the germline, what gets passed on to you, and the diversity really gets generated in your own B cells, so at the level of somatic. And so really you're going to get somatic recombination events of segments within your heavy and light chain. And those segments are referred to as V, D, and J. So this is variable, this is diversity, and this is joining. Now, the recombination events here are thought to have the capacity to make about 10 to the 11th different antibody or different immunoglobulin with different specificities. So the recombination events at the somatic level are really going to give you the breadth to respond to the pathogens that you might encounter for your life. Now, after you get these recombination events, you get your heavy and light chain polypeptides. And then these, of course, will form your immunoglobulins. Okay, so let's talk about the immunoglobulin genes. And so with respect to your immunoglobulin genes, you are going to have heavy chain and light chains. So we'll start with the heavy chain. And we're just talking right now about how we get diversity. So I'm not going to include all the constant region genes focusing on the diversity. So we have our heavy chain. And your heavy chain is going to be on chromosome 14. And so you're going to start your heavy chains with what we refer to as leader sequences, these L sequences. And the leader sequences are important in exporting your chains, heavy, light. Light will also have them from your nucleus to the ER, where they can become proteins. So that's what these leader sequences are for. So you'll have a leader sequence, and the leader sequence is always right in front of your variable heavy chain. So right after your leader sequence, you'll have a variable H1. So this, we'll just leave that alone, variable H1, and then you will have another leader sequence, which is going to be in front of variable H2. And this will continue. And so in the human genome, we have 65 variable H regions. So here's our leader. So variable H65. And so you'll have 65 of these, which will always have a leader sequence, then your variable region sequence. And then after you have your variable regions, you have your diversity genes. And there are 27 known functional diversity genes. Oh, like that was purple. So your diversity genes do not, have a leader sequence. And so you have diversity 1, diversity 2, diversity 27. And then after your diversity genes, you will have your joining genes. So you'll have your joining 1, joining 2, all the way out to joining 6. Okay, so you have 65 potential variable heavy regions. You have 27 potential diversity regions and 6 of your joining regions. And so these are going to combine to form the variable regions with a breadth of abilities to bind to different pathogens on the heavy chain side. Now we also have our light chains. So your light chains, you have 2 light chain segments. You have your kappa light chain. And your kappa light chain is on chromosome 2. And then you also have another light chain. And that is a lambda light chain. And your lambda light chain is going to be on chromosome 22. Okay, so your kappa and lambda light chains are going to have your variable regions similar to your heavy chains. They have a different number of them. Your kappa variable region is going to be 40, and your and so you're going to have 40 potential variable regions, and then you're going to not have diversity genes. So let's see if I can just do this. And so they don't quite have as many. So we're going to put on here 30. And you don't have any diversity genes. And so with your kappa, you're going to have your joining genes. And there's going to be potentially 5 joining genes. Okay, and then you're also potentially going to have your lambda light chains. So you can have variable regions on your lambda, which can be up to 30 variable regions. And then you also, with your lambda, have just a single joining region. And so that's different than your kappa chain. Oops. Sorry about that. And this one can potentially have about 30, so a little bit less near kappa. Only has one joining. And again, no diversity genes. And so this is your heavy chain, your kappa light chain, your lambda light chain. And so I wrote in this order because this is the order in which they get transcribed. So first up will be your heavy chain. You do heavy chain rearrangement, followed by your kappa chain rearrangement, and if necessary, you will go to your lambda gene rearrangements. And I say if necessary is because your kappa is always first, and if you get a successful antibody with your kappa, you're not going to go and rearrange your lambda gene. Okay, so now let's talk a little bit about how this all works. And so it's pretty straightforward. You're going to have first, as I said before, your heavy chain's going to rearrange. And so the first thing that your heavy chain has to do is it has to choose a diversity and joining gene. And so first you have your germline. And I think I'm going to go back here and steal this. Okay, so here's your germline. And then from this germline, you're going to get a diversity and joining gene come together. And so you will have, for example, you will have still your variable regions. You haven't yet chosen a variable region. And your D and J joined or rearranged and came together. So you picked a diversity gene. We'll say we'll pick diversity 5. And then we picked a joining gene. And it can be joining 1. So we have D5J1 now. And then upstream from all of this, which I didn't put in the previous slide, is what we refer to as our constant regions. So I'm just going to put a big block here. And we'll talk about that in just a moment. Okay, so now we have our recombination between our diversity and joining genes. And then we're going to get a recombination of those genes together. And then we're going to get your variable region joined together. So in this case, we have our leader sequence. Always goes with a variable region. We have our variable region. We will pick variable heavy 20, and then we have our diversity 5. We have D5 coming in, and then we have our joining gene, which is 1, joining 1. And so now we have a recombination, our second recombination. And now we have our leader sequence, we have our VDJs together, and we have our constant region trailing behind. And so we're going to get transcription of our heavy chain transcript. And of course, transcription is to make your messenger RNA, obviously. And so your messenger RNA is going to undergo what messenger RNAs undergo, and that's going to be splicing. So all of these chunks here, here, here get spliced out, for example, and then you get your messenger RNA. And the messenger RNA, of course, is going to have our leader. It's going to have our variable region. It's going to have our diversity gene. And it's going to have our joining gene. And it's going to be followed by our constant region. Okay, so there's our messenger RNA. And then the messenger RNA is going to be transported using the leader sequence to make your proteins. And your proteins are going to eventually pair up with your light chain, and then you are going to get your immunoglobulin made. Okay, so some things to keep in mind with the heavy chain constant regions. Okay, so our constant regions are going to be what kind of identifies your heavy chain. So remember, we have different antibodies. We have IgM, which is also known as mu. We have IgG, which is our gamma. We have IgE, which is our epsilon. We have IgD, which is our delta. And we have IgA, which is our alpha. Oops. Alpha. Okay, so constant regions are going to be set up with those particular genes. So for example, I can add to these constant regions. Let me just make this longer. We're going to have the mu, we're going to have the delta, followed by our gamma. And gammas have many different varieties, so there's several different gammas. Won't go into that right now, but I'll just draw a few gammas, 4. And then after the gammas, we'll have our epsilon. And then after the epsilon, we'll have our alpha. Now, your antibodies are always, as I mentioned before, always going to start out with a mu. So the first antibody ever made is always going to be the IgM. Okay, and so that's where it's really going under constant region. That mu is the first one after your joining gene, and so that's going to be expressed more. The next one you can see is the delta, so that will be expressed also, and we'll discuss that at a later time. So now what happens with the development of your light chain? So again, your light chain, you have, again, your germline, and you're going to have, once again, your leader sequence. And then you're going to have your variable regions. And you're going to have your joining regions. Oops. I'm going to keep the same color. Yeah. Joining was blue. And you're going to have your constant region. So the first thing that's going to happen in your light chain is you're going to get recombination between your variable and joining. So again, you have your leader. Now you have your variable, and you have your joining coming together. And you, of course, have your constant region still there. And you get your primary messenger RNA transcript. And you, of course, have to splice out your introns. And so you get your messenger RNA, which will be similar to your heavy chain. You have your leader, and now it is right next to your variable, which is now right next to your joining and right next to your constant region. And your messenger RNA will then go to your ER and will be transcribed or translated into a protein. Okay, so a few things about the heavy chain and light chain, just to explain what's going on with your constant regions and why it's different if you're a heavy chain or a light chain. So how you can imagine your antibody is, again, your antibody is this Y-shaped. So you have your little arms here. And you have another set of them over here. Okay. And then you have your trunk. Now, every antibody has a little bit of different structures in what we call the hinge region. So I'm just going to draw this kind of as little squiggly lines. So this area here is referred to as the hinge, and it provides flexibility. Also, you're going to have variable regions and constant regions, and so that's where these constant and this VDJ or VJ regions come in. And so on your heavy chain, you're going to have your variable heavy. And on your light chain, you're going to have your variable light. And then for your constant chains on your light, it's just simply constant light and a constant light. And in your heavy chains, they're given different sections. So the section that's closest to your variable region on your heavy chain is called constant heavy 1. And so that's this area and this area. And then you'll have two other constant regions. You'll have constant heavy 2 and constant heavy 3. And of course, it'll be the same on this side. Okay, and so that's sort of the structure of your antibodies. The antigen, of course, will bind here. So the antigen binding is here. And down here, oops, it's a horrible squiggly, is where the function of your antibody is. Let's write it this way. Okay, so different antibodies have different functions. Some are going to activate complement, some are going to cross the placenta. That's better. And so it just depends on what antibody is, what function it will have. Now, just very quickly, so B cell receptor versus a actual antibody. So one thing to keep in mind is that the B cell on the surface has a B cell receptor. And the antibodies are going to be secreted. Now the specificity is exactly the same. So what I mean by this is if on the B cell surface, let's do a B cell. The B cell has an antibody on its surface, and it's specific for a square, a red square. This same B cell, when it secretes an antibody, it is still specific for that same red square. So the specificity never changes. What's changing is whether or not you're going to get a secretion sequence or you're going to get a membrane embedding sequence. And that's going to happen at the primary transcript level. So one way to think about this is prior to activation, they are all going to be B cell receptor. After activation, you're going to have B cell receptor or antibodies. Okay, so this is a B cell. This is a B cell. Now, if the B cell becomes a plasma cell, this is only going to be antibodies. Plasma cells do not express B cell receptors. They are simply just antibody making machines, and that's all they release are antibodies. And so basically in your messenger RNA, you have membrane embedding sequences that we didn't add. So just very quickly, we can add that. So in our transcript, we had, of course, our leader. And then we, of course, had our variable region, diversity, and joining all together. And then we had our constant region. And then after the constant region is another area which you have basically polyadenylation sites and transmembrane sites. So I'm going to draw a poly A site here. And another poly A site here. You see they have two poly A sites. And then, let's see what color. We have our transmembrane sites, TM1 and TM2. And so remember a gene, any gene will have, so here's just any gene, will have a 5' cap with the gene, and then a poly A tail. And the poly A tail really tells that this is the end of your gene. And so what we're showing, what's going on here in this section over here is you have two different poly A tails. So if you get spliced here, then this means you get an antibody. If your splicing is here, you're going to add one of those transmembrane domains, and you're going to get a B cell receptor because you're going to get that membrane domain saying, I want you to embed into the membrane, which means you're a B cell receptor. If you don't have that, you're an antibody and you get released. Okay, so moving on, let's talk a little bit about diversity. So what are the key points of antibody diversity and how is this diversity explained? Well, there are multiple points to diversity within an antibody. The first point is your germline diversity. And this came from having multiple copies of your VDJ. And you get one copy from your mother and one copy from your father, so that adds to that diversity. The second is called combinatorial, or combining. And this is where you have VJ or VDJ for heavy chain recombination events. And remember, this is somatic. And then you also have what we call junctional diversity. And the junctions that form between gene segments, such as when your V and DJ genes are joined together, creates variable joints with different coding sequences. And then we also have random assortment. And any heavy chain can associate it with any light chain. And then finally, something we haven't discussed yet, but we'll discuss in a moment, we have somatic hypermutation. And this happens only after an antibody binds to antigen. The antibody can get better. And we'll discuss that in the future. So up to this point, everything is happening prior to antigen. And this is after antigen binds. Okay, but all of these have to do with the diversity of your B cells and your antibody repertoire. Now, the last thing to talk about with respect to this is class switching. Okay, so again, when you have your gene, and this is only going to be on the heavy chain, the light chain doesn't do this. So when you have your gene, you have your V, make sure I do the right color here. Okay. D, and then J. And then your antibodies have to always make the mu constant region first. So here's our mu chain. And the mu chain will have constant region 1 mu, constant region 2 mu, constant region 3 mu, etc. And so the mu would give you an IgM. The next one down is the delta. And so this combined with this, let me do a different color, to dark green, like delta, dark green, delta. So this combined with this will give you IgD. And so both of these together will be on the surface of a naive B cell. Now, naive means never seen antigen. Okay, and so we have our naive B cell. And on the surface, it's going to have an IgM. And on its surface, it's going to have IgD. So IgM and IgD. IgD has no function, and so the only time you ever see it is when you have a naive B cell. So you know you have a naive B cell if there's IgD on the surface. Now when a B cell gets activated, you have to do isotype switching. Okay, and so our isotypes are, we have IgG, we have IgA, and we have IgE. So IgM must choose, am I going to switch to IgG, IgA, or IgE? And so if you're going to be in a secretion, such as in the gut, you can switch to an IgA. If you're going to have a pathogen, so an intracellular or extracellular pathogen, bacteria, you go to IgG. If you're going to be a parasite, extracellular, or if it's an allergic response, you're going to become IgE, and you're going to undergo isotype switching to do that. And so for isotype switching, you're basically, again, I can take our little gene up here, and your body is very clever about this because it put on purpose IgM and IgD first. Oops, sorry. And there's a reason for that. So behind IgM and IgD, let's just say we're going to become an IgG. So here's our gamma. And this one was our delta. So we have our mu, our delta, and our gamma. And we decided we're going to switch from our IgM or our mu to our gamma. So what's going to happen is that you are going to get splicing of your transcript here and here, and then your transcript is going to, and also here, your transcript is going to become, oh, sorry. Well, that just didn't work. Let me just draw it. Your transcript is basically going to become VDJ I put gamma as the same color, sorry about that, and I'll make it bigger with your gamma. And so this will always be an IgG producing B cell. Okay, and so now it's lost its ability to make the M, it's lost the ability to make the D, ability to make the E, A, etc. It's just simply I'm an IgG producing B cell now. And that's called isotype switching, and it happens after activation. Okay, so that's all I had for antibody diversity.